All right, guys, let's talk about this remarkable metabolic pathway known as the citric acid cycle, right? This amazing sequence of events, right, um, is known as the citric acid cycle in principle because of the central role that citric acid plays in it, but it's got some other names too. It's also known as the tricarboxylic acid cycle, or the TCA cycle for short, right? Um, and the basic outlines of the cycle go something like this, right? And I'm going to divide the TCA cycle into sort of three phases. It helps me kind of remember what's going on, right? So we're going to start out, again, as I said before, the input into the TCA cycle is, of course, pyruvate, right? So that alpha keto acid that we got out of the back end of glycolysis, right? So let's say, glycolysis is coming in here. This is what's feeding our pyruvate pool, right, primarily. Okay, at least in thinking about carbohydrate breakdown, right? So the first phase of this cycle, right, and what I'm going to call sort of phase one, is a set of steps. It actually happens in one complex, which we're going to see in the sort of next set of descriptions. I consider this sort of phase one of the cycle, where we're going to take some NAD plus to create some NADH, and we're going to drive some CO2 off of pyruvate to generate that first of those sort of one of those activated carriers that we were talking about the last time, right? Essentially, we're going to use this oxidative decarboxylation reaction to activate those two carbon units, that acetyl group, right? That's in pyruvate, to create this thioester, activated thioester, which we talked about before, of acetyl-CoA, right? All right. All right, that's phase one of the cycle, all right? Phase two of the cycle, all right, that's really sort of the entry point. That's sort of just getting us set up because we need those activated acetyl groups to really get things going. And where this happens, or what happens now, is we're now going to take a dicarboxylic acid, known as oxaloacetate. Oops, let's draw this a slightly different way. Oh, come on. Yeah. All right, so this is oxaloacetate, all right? And don't worry about it for now. We're going to come back to it. And the first step of the cycle that really gets us going is a condensation reaction between acetyl-CoA and oxaloacetate to form that first of the tricarboxylic acids, right? Which is this guy here. Which is, of course, citrate, right? That key molecule for which the cycle gets its name, right? Or one of the key inputs for which the cycle gets its name, right? We're then going to run, and this, I consider this all sort of part of phase two of the cycle, right? So if that first step, that creation of acetyl-CoA, right? If that, we're going to consider that sort of phase one of the cycle. Phase two of the cycle begins with this formation of citrate, this condensation of acetyl-CoA and oxaloacetate to form citrate. And then we're going to run through a series of oxidative decarboxylations, right? Not unlike the one we just ran, where we're going to like create some NADH and throw off some CO2, right? We're going to do this a few times, right? Or a couple of times, I should say. All right, some more CO2, right? To generate another thioester containing compound known as succinyl-CoA. All right. So we're going to use CoA again. Right. Right, so we're going to launch a couple of CO2 off, those CO2 groups and citrate off to create succinyl-CoA and a whole bunch of NADH, right? So this is, as I said, succinyl-CoA gets us kind of halfway around, right? So all of that is what I call phase 2 of the citric acid cycle, right? Okay. Phase 3 of the cycle, we're basically now going to run a series of steps designed to get us back up here to oxaloacetate. One of these steps is actually going to make some ATP, or depending on the isoform or the cell type, GTP. It's the only substrate level phosphorylation event that happens in the TCA cycle. And then these other steps are designed to do some isomerization and suck more electrons out of the structure of this molecule as we convert it back, what's left of it, back into oxaloacetate. And the second phase, from succinyl-CoA back up to oxaloacetate, I sort of think of then as phase 3, or I like to think of as phase 3 of the cycle, right? Okay. All right, and that brings us full back around in this cycle to where we can then recondense, right? So you can sort of see what's going to happen here is the carbon input from succinyl-CoA and oxaloacetate are kind of going to treadmill through this cycle as we extract electrons and make a little ATP. That's sort of the big picture goal. Now, you know, there's another name for this cycle. It's probably worth knowing. This is sometimes also known as the Krebs cycle, right? For Hans Krebs, the chemist in the 1930s, who was the person who first started working on this and really mapped it out. That the sort of this cyclic reaction pathway that generates CO2 and water. It's an idea for which Krebs eventually won the Nobel Prize, though not without a lot of resistance. Remember, this is in the 1930s. Krebs is working on this at about the same time that Eigen and Emden, Meyerhoff, and Warburg were working on glycolysis, and that's a very clearly linear pathway. And people, I think in general, were thinking of these pathways as linear transformations. And what Krebs proposed here was something radically different, right? This sort of a very unusually arranged pathway. And he was working in an odd system. He was working with minced pigeon muscle. Don't really know why, other than maybe, you know, living in a city and there are lots of pigeons and so readily available source. But I don't know, minced pigeon muscle was his model. And he noticed that when he minced up pigeon muscle and made a lysate out of it, and he added certain organic acids, citrate, isocitrate, sisaconitate, and then monitored oxygen consumption by these lysates, the oxygen consumption would burst in direct proportion to the amount of the citrate or isocitrate or sisaconitate that he added, but the oxygen consumption would exceed significantly the mass of the citrate or isocitrate he put in, as if the these tricarboxylic acids were acting catalytically, right? And that was really kind of a radical idea for the 1930s. People had a hard time sort of getting their brains around that, but Krebs is right. And in fact, the cycle that I'm sort of indicating here that Krebs mapped out, he got almost exactly right with the tools in the 1930s. The cycle we know today as the TCA cycle, or the citric acid cycle, is almost exactly, with only a couple of modifications, the cycle that Krebs proposed, right? And like I said, he eventually won the Nobel Prize for it. Okay, so the TCA cycle is, of course, this central engine right here at the core of intermediary metabolism. It's sort of the furnace, right? If glycolysis was one of our big inputs here, coming down here, we got polysaccharides, monosaccharides, convert anything we can kind of convert to glucose, we can just pump through glycolysis, and then on into pyruvate. The TCA cycle, man, anything that you can run down through this pathway into pyruvate or acetyl-CoA, you can burn into the TCA cycle. In fact, even though, like I said, we can get energy out of monosaccharides, you can even get energy through glycolysis out of amino acids. So a subset of them can be converted into monosaccharides and then run through glycolysis. But when you really want the big payoff, you really want ATP, you want to burn for maximum energy, and that means amino acids, from proteins, carbohydrates, or even from lipids. Your maximum energy, your goal is maximal energy production. The furnace you're going to use is the TCA cycle. And there are multiple ways to get things going into the TCA cycle, either through glycolysis. You can come in sideways through acetyl-CoA, which is how fatty acids are pumped in. And you can even run, as we'll see, many of the amino acids sideways into the TCA cycle and burn them directly in the TCA cycle. Just about anything that being turned into an acetyl group or a dicarboxylic acid can be combusted in the TCA cycle for energy, right? Now, a corollary of this is that the TCA cycle, like we saw with glycolysis, is really important for intermediates, right? Right. So the TCA cycle is run not only for energy, right, but it's also run to get metabolic intermediates that are necessary for synthesis for biomass, these so-called anaplerotic, right, reactions, where you run the TCA cycle less for energy, more because there are intermediates you need for doing biosynthesis. So the TCA cycle is important for both things. It connects in a really big way energy production with anabolism, really key important elements of anabolism. Okay, now there's a cell biology wrinkle we should bring in here, which is that hopefully you remember that glycolysis, the TCA cycle, are not happening in the same place, right? If you remember that all the enzymes of the glycolytic pathway that we've been talking about, they're out in the cytoplasm, right? And people used to think these were just isolated enzymes floating around and the substrates bounce into each other. There's growing evidence that that's probably wrong. Large numbers, maybe not all of, but many of the glycolytic enzymes are probably involved under metabolically active conditions into large supercomplexes. Some people call these glycosomes sometimes. I believe that's the term I've heard bandied about, right? But all those enzymes and all that chemistry we just talked about in glycolysis is happening out of the cytoplasm, all right? Now, in bacteria, TCA cycle, citric acid cycle, TCA cycles, going to be happening in the same place, and there's an important need to segregate. But in eukaryotic cells, right, organisms like us, any and all multicellular or even just eukaryotes, right, glycolysis and TCA cycle are segregated from each other because the TCA cycle is running in the mitochondria, right, which sets up a really interesting issue that all that pyruvate that we've just made in glycolysis has to get over here into the mitochondria. And it does, but here's the interesting thing. Mitochondria have no pyruvate transporter. So all that pyruvate that's got to get in here, that is the TCA cycle, not totally, but substantially dependent on, has to get into the mitochondria, and there's no way to directly transport it. And there's some fascinating indirect ways in which these pyruvate equivalents are moved into the mitochondria, along with some reducing equivalents, right? And we'll talk about that as we go along. Okay, so glycolysis in the cytoplasm, TCA cycle in the matrix of the mitochondria, right? Okay, and just as sort of a big picture overview, again, I gave you my sort of three-phase overview. There are a couple other ways to think about it. Sometimes people like to think about this as sort of the two phases, or take pyruvate, make acetyl-CoA. That's our initiating step. And then we run the TCA cycle. And sometimes it helps just thinking carbon equivalents, right? So that acetyl group that's coming in is a 2-carbon unit. Oxaloacetate is a 4-carbon unit, right? Dicarboxylic, 4-carbon dicarboxylic acid. Condense those guys to make a C6, that tricarboxylic citric acid. And then we're going to treadmill this guy around, throwing off a couple of CO2. So we're going to go from a C6 to a C5, then to a C4. And then it's a matter of some more oxidation steps to get this baby back up to oxaloacetate. right? To do this is going to take, again, about 10 steps. So in the total number of reactions, it's similar-ish, depending on how you categorize it, to glycolysis, right? And to come all the way around to drive the TCA cycle. And that's what we're going to dive into next time.